So the first thing we're going to establish, guys, is why do all cells need to respire? All right, that's actually one of the features of living organisms. Good morning, good morning. So all cells must respire to produce ATP, which stands for adenosine triphosphate. In order to carry out cellular functions. So every single cell must be able to do the process of respiration to produce the molecule of ATP, and this allows it to carry out its cellular functions. And you might be wondering, what are some of the functions that the cell requires energy for? So let's look at some of the processes that the cell needs energy for. So we say all organisms require energy for survival. Energy is required for many reasons, including one, they need energy for anabolic reactions. And that's when you're building up molecules. So whenever you want to build up a molecule, that's an anabolic reaction, you need energy for that. You also need energy for catabolic reactions. So we also need energy for catabolic reactions. Or let me do that better. We need energy for movement, for reproduction, to maintain body temperature, and to transmit nerve impulses. These are just some of the functions of energy that all living organisms need it for. So anyway, the whole point of what I'm trying to say is that every living organism needs energy, or every cell needs to have its own supply of energy. You can't get energy from a neighboring cell. You have to do the process of respiration for yourself to generate your own ATP. So, and I'm going to look at how energy flows. So the ultimate source of energy is the sun. which provides us with solar energy. And then we have the leaves, which capture the solar energy, converting that into glucose. The glucose contains chemical energy in the bonds of the molecule. And that energy is harnessed by cells, which basically are able to release the chemical energy in the glucose. So in the cell now we have the mitochondria, right? So basically the glucose goes into the mitochondria or mitochondrion, and then that produces ATP. So the aim is to show you that ultimately the ATP that we are making is actually coming from the sun. It was sourced from the sun. Now, so we see that the solar energy that we captured during photosynthesis eventually will be converted into chemical energy. And that chemical energy will be used, will be captured in the ATP molecule, which will be used for all the reactions that we need to do in the cell. And I gave you like 6 examples above that we use the energy for. I'm going to tell you some more about this molecule of ATP that you're going to see very commonly. So what we're seeing in this flow chart here is that we're seeing solar energy being converted into chemical energy. And the chemical energy in glucose is then released during the process of respiration. The energy that is released from the glucose is captured in the bonds of ATP. And whenever a cellular process requires energy, it gets the energy from the ATP. So ATP is basically the immediate source of energy for basically all cellular processes. But the energy originally came, the energy that's in the ATP originally did come from glucose. So we say that ATP is the immediate source of energy, and glucose is considered as the short-term source of energy. So I'm going to tell you a little bit more about this molecule here of ATP. More about the molecule of ATP now. This adenosine triphosphate molecule. So it's actually a nucleotide. So it's made up of a ribose sugar with a base, an adenine base attached to the ribose sugar, and 3 phosphate groups. The bonds between the phosphates are the bonds that we usually break. So if you should break the bond between the 2 last phosphate groups, you can get 30.5 kilojoules of energy from breaking that bond. And if you go between the first 2, you can get 31 kilojoules of energy. Let me tell you the energy for the next one. Yes, I'd like you to copy the diagram, please. Question, do they normally ask about the amount of energy you mean in the entire ATP molecule, or you mean in each bond? No, they won't like ask you directly like that, but you need to know that each bond contains energy. So when we say that, so we usually tell you the overall energy in ATP, but it's helpful to know the bonds, to know that the most of the energy actually comes from the first 2 bonds. But most times you only break off 1. But if you need it, you could break a second one. So I'm trying to find the energy of the 3rd bond. So I guess I'm only seeing the first 2 because most times the molecule doesn't try to break that 3rd bond here. I think I see. I think it's like -20. It's like 20.5. So one glucose molecule, if you should burn an entire glucose molecule, you could get over 2,880 kilojoules of energy. All right. Now, what the cell does is that it captures that energy and makes ATP. So basically, ATP has roughly 80 kilojoules of energy for each one, 70 odd or 80 kilojoules for each one. So we can make up to 34 ATP molecules whenever we burn one molecule of glucose. Now, the good thing about ATP is that ATP stores the energy in smaller packets. So it's basically storing it in packets, and you can actually use the entire molecule of ATP, or you could literally just break one bond. So if you have a process that only requires like 28 kilojoules of energy, then you literally can only break one bond of the ATP and you get roughly that 28. So what this does now, by storing the energy in ATP, the cell has a better, it has more efficient use of energy, because a lot of the processes do not require thousands of kilojoules. They require like 10 or 20, 30 or 200. And basically, the cell can just calculate how many ATP molecules it would need to meet that specific cellular process's need. And so it doesn't waste energy. So whenever the cell has a process to do, it doesn't use the energy from glucose directly. It uses the energy from glucose to make ATP, and then it calculates how many ATP molecules it requires for the process it needs to do, right? So ATP is like the energy currency of all cells, and it allows the cell to be more efficient in its use of energy. So we're actually going to talk about, in a moment, we're going to compare ATP's features as compared to glucose as an energy source. So water is needed to release the energy from ATP in a process called hydrolysis. So if we add water to our ATP molecule and we break off just the last phosphate group, then we form ADP, which is adenosine diphosphate. And if we have ADP, we add water, more water, we can form AMP and PI and a phosphate group. And of course, energy is given off as well. So the bonds of ATP can be broken independently. And this is why ATP is the immediate and source of energy for cellular reactions. Because it can hydrolyze each bond independently. According to the amount of energy required by the process. So ATP is the immediate source of energy. We say glucose is the short-term source, but ATP is the immediate source. So we're going to make a comparison table when you're done, showing you if we should compare the features of glucose versus ATP. So we're going to compare ATP versus glucose. All right, so I'm going to speak first about the benefits of ATP and then speak about glucose afterwards. I want to look at some features of ATP. And then I'm going to look at the benefit of the feature. And then I'm going to discuss glucose. So the first feature that we will speak about for ATP is that it releases a small amount of energy. So in total, it's about 75.8 kilojoules per mole for each molecule. So if we were to break all three bonds, we get about 78.5. This is a good feature because this is, to drive most metabolic reactions. And it keeps the energy wastage low. Remember, glucose is the immediate energy source. So this other column is for me to discuss what if we had been using glucose as an immediate energy source versus ATP. We'd see that glucose would not be good as an immediate energy source. So glucose gives 2,880 kilojoules per mole, which is in excess of what is required for most metabolic reactions. A lot of energy would be wasted as heat. So if we had chosen glucose as our immediate energy source instead of ATP, we would have been wasting a lot of energy as heat. We don't get that wastage with ATP because ATP gives a small amount of energy, which is closer to what most reactions would require. The other thing is that ATP exists as a stable molecule. And this benefit of this is that it doesn't break down unless a catalyst is present, which is ATPase. And so energy won't be wasted. So ATP is not just going to get up and break down like that unless the catalyst is there. We don't really have a matching point for glucose for that one. ATP can be recycled. Because the breakdown of ATP is a reversible process. So it's a ATP can break down to give ADP and PI, but you also could go around reverse it and put back the ADP and the PI together, the phosphate together, and get back the ATP. So this means the same molecule can be reused. So the same molecule can be reused in the cell. On the other hand, glucose is not recyclable. So we have some other features. So when you want energy from ATP, it's a very quick process. I really should make this category bigger, guys. Sorry for the untidy table, but this benefits category should have been bigger than how I drew it. So whenever we need energy from ATP, the hydrolysis is quick and easy. And what benefit is that? This allows cells to respond. To sudden increases in energy demand. On the other hand, to break down glucose is way more time consuming. It occurs in 3 stages and multiple bonds must be broken. ATP is soluble and moves around easily within the cell. And the benefit of this is that it can transport energy to all areas of the cell. And to glucose's credit, it is also a soluble molecule that could go anywhere in the cell as well. So that's a feature that glucose has, which matches up with ATP. ATP forms phosphorylated intermediates and this can make metabolites more reactive as it lowers the activation energy of the reaction. So for example, ATP can react to the substance and a phosphate group is added to the substance. And this makes the substance more reactive than if when it never had the phosphate group. Glucose cannot do that. Finally, ATP cannot leave the cell. Because it cannot leave or enter cells. So it means that each cell must maintain its own constant supply of ATP. On the other hand, glucose is free to enter and exit cell. All right. So remember that it's the energy from glucose that gets captured in ATP. So our reactions in our cells, they don't, whenever the cell needs energy, they don't reach for glucose directly, they reach for ATP as the immediate molecule of energy. And then the cell burns glucose in order to replenish the ATP stores. So that's the order that it goes in. Scroll back here. Okay. All right, so let me give you a chance to finish this, guys, as we learn more about ATP. Right. So I'm going to be showing you a video about the features of ATP in a moment. In another minute, guys, I'm going to show you the video just to reinforce the features of ATP. So let's look at ATP.
videos. So first of all, ATP is in the biological. Hi and welcome to an A-Level Biology for Free with Miss Estrick. This video is going to go through ATP for A-Level Biology. If you are new here, make sure to click subscribe to keep up to date with all the latest videos. So first of all, ATP is in the biological molecules topic and it is a nucleotide derivative. And that is meaning it is made up of many of the same structures or molecules that a nucleotide is. And ATP stands for adenosine triphosphate, and its function is as an immediate source of energy in metabolism or biological processes. So it's essential that all cells have a constant, steady supply so that they can undergo all of the chemical reactions which have to take place in the cell. And that release of energy is linked to the three phosphates, and we'll have a look at that in a bit more detail. So first thing you need to know is the three components that ATP is made up of, and that is adenine, which is a nitrogenous base, and what we mean by that is it's a base which contains nitrogen, and in this diagram here you can actually see where the nitrogen is. It also has a pentose sugar, which means a 5-carbon sugar. We can see that 1, 2, 3, 4, 5 carbons. And the pentose sugar that it contains is ribose. The final structure is the 3 phosphate groups. And these are inorganic phosphate groups because they do not contain any carbon atoms. So you can see here it just contains phosphorus and oxygen, which is what a phosphate group is. So sometimes in a word equation or a symbol equation, you might see it's shown as just a P or Pi to show that it's inorganic phosphate. Now for AQA, the level of detail that you'd need to be able to remember that structure in is just the shape. So you don't need to know any of these individual atoms. You just need to know you have an adenine base, ribose sugar, and 3 inorganic phosphates. So ATP is made during respiration. So you'll have ADP, which is adenosine diphosphate, which if I just go back to here, you can see that is highlighted here, where you would only have 2 of the 3 phosphate groups attached. And so in respiration, a condensation reaction occurs whereby 1 inorganic phosphate group is added to ADP, and that is using the enzyme ATP synthase. And that does take a little bit of energy to do, to add and create that bond. Now the opposite is how ATP releases energy. So once you have ATP created in respiration, you can hydrolyze one of those bonds between the phosphate groups to create ADP and PI. And this also needs an enzyme, and it's ATP hydrolase, and this is the step where you do get a small amount of energy being released when that bond between the phosphate group is broken or hydrolyzed. And it's described as an immediate source of energy because in order to release that small amount of energy, only 1 bond had to be hydrolyzed, so you get this energy very, very rapidly. So this diagram here is just representing it is a reversible reaction. For ATP hydrolysis, you have to add water and you need ATP hydrolase. The reverse of that reaction is how ATP is created, and this is a condensation reaction because water is removed, and ATP synthase is required for that reaction, as well as a small amount of energy. So the final thing just to point out is ATP can transfer the energy that is released when that bond is hydrolyzed. So instead of it just being released to the surroundings, if the inorganic phosphate, which has been broken off, is bonded onto a different compound, that compound will then gain the energy and it becomes more reactive. And that is known as phosphorylation. And this reaction here, ADP and PI joining together to make ATP, that is phosphorylation because the ADP is becoming more reactive and it's gaining energy by adding a phosphate group on to become ATP. And phosphorylation is also the first stage in respiration, in glycolysis, which is in aerobic and anaerobic respiration. So the final thing is looking at some of the key properties of ATP, which makes it different to glucose as an energy source. So it's an immediate energy source, as I've said already, and that's one of the key differences between ATP providing energy for a cell instead of glucose. But you could get exam questions asking to compare ATP to glucose in providing energy for a cell. So the first thing to point out, and this emphasises why ATP is more suitable, it only releases small, manageable amounts of energy when that bond between the phosphate groups is broken. Whereas if you were to break down glucose, you'd get large amounts of energy. The reason this is useful is you're going to be wasting less energy because you will only release small amounts. If that's not enough, you hydrolyse more ATP until it adds up to the quantity that you need. So it prevents wasting energy, and also a lot of wasted energy is in the form of heat, so it stops the cells from overheating. Number 2, ATP is a small and soluble molecule, so it can easily be transported around a cell. Now that is actually the same as glucose. Glucose can also dissolve in the cytoplasm and transport around the cell. And this is useful for both molecules because wherever they are created, so glucose might be absorbed, but ATP is created in the mitochondria perhaps, it can still be transferred to other parts of the cell for metabolism. Number 3, only one bond has to be hydrolyzed for ATP to release energy, which is why it's an immediate source. Whereas glucose, you'd have to hydrolyze several bonds, so it'd take much longer for it to release all of its energy. Number 4 is this idea of phosphorylation that we referred to. So with ATP, when you do hydrolyze the bond between the phosphate groups, that inorganic phosphate can be then bonded onto a new molecule and make that molecule more reactive. And you can't do this with glucose. The final thing is ATP cannot pass out of a cell. That means that it can't diffuse through the cell surface membrane, and there are no protein carriers which it can attach to. So every single cell has its own supply of ATP or ADP and PI, whereas glucose can move in and out of cells through channel and carrier proteins. So a cell can run out of glucose, but a cell will always have a constant supply of either ATP or ADP and PI. So that is it for ATP.
Right, guys. So I had wanted to just hear it from another perspective, why this molecule of ATP is so suitable for its use, which is basically to act as the immediate energy source. And you saw the features. And of course, this is something that Kate loves to ask you. So we're going to start looking at how do we actually manufacture ATP in our cells? How do we make it? So we know the ATP is made from the energy that comes from glucose, but what exact process do we use inside of our cells? So we actually have two ways to make ATP in our cells. We're going to be exploring both of these methods today. All right, so in order to make ATP, we make ATP during two reactions. We make ATP during photosynthesis, if you're looking at plants, and also in respiration. But if you're an animal like us, all of our ATP comes from respiration because we can't do photosynthesis. Now, ATP can be made in one of two ways in our cells. There's one process called substrate-linked phosphorylation or substrate-level phosphorylation, and then there's one called chemiosmosis or oxidative phosphorylation. We actually met the chemiosmosis one already during photosynthesis. So we're going to meet it again, right? So I'm going to ask you to go ahead and see how we make the ATP. We're going to get into both of these processes, starting off with the substrate linked technique. So guys, we're gonna go through substrate level 1 first, and then I'll give you a break, and then we tackle the second one, the chemiosmosis. All right, so substrate level phosphorylation is we're basically transferring a phosphate group from a different molecule to the ADP. So let's look at that substrate level phosphorylation. ATP is formed by transferring a phosphate group directly from a substrate to a molecule of ADP. So literally, we have the molecule of ADP and we have a substrate, which is basically a molecule that has a phosphate group attached to it. And then the phosphate group is given to the ADP to make ATP. And then we have the substrate losing the phosphate group. So that's literally how it works. So the energy to make the ATP this way for this reaction is provided directly by another reaction. And this process usually occurs in the cytoplasm or the matrix of the mitochondria. This only accounts for a small amount the ATP synthesized during aerobic respiration. That is approximately 4 to 6 molecules per glucose molecule. This method is also utilized during glycolysis. And requires an enzyme. So basically, we're just taking the phosphate group directly from one substance and adding it into the ADP to make it into ATP. And that doesn't make much of the ATP in our cells, though. Only about a very tiny fraction of our ATP is made by substrate level phosphorylation. So it means that the bulk of the ATP is going to be made by the second process, which is chemiosmosis, and that's the one that we're going to focus on. So I'm going to give you a break after you finish it. I'll give you 2 more minutes. Give you a break. And after the break, we're going to continue with chemiosmosis. And we're also going to be learning about the structure of the mitochondria. So those are 2 other ones we have to do. Guys, so I'm going to give it a break now. After the break, we're going to continue learning about chemiosmosis and the mitochondrial structure. All right, guys, we're going to look at the second method now for making the ATP. This is known as chemiosmosis, and as explained to you that we actually met this process already.
All of the steps.
Met it already when we're doing photosynthesis. So we're going to see it again in respiration.
Of aerobic respiration that we have done so far have been leading up to this, the electron transport chain. This is the main event, the big bonanza, the Super Bowl of cellular respiration. Why? Because the vast majority of our ATP is made here. The electron transport chain is actually a series of proteins and other molecules embedded in the inner membrane of the mitochondrion. These molecules transport electrons, hence the name. Let's zoom in on this inner membrane to see the electron transport chain in more detail. Here's how it works. NADH, that electron carrier we've been making all along, delivers its electrons to the first protein in the electron transport chain. As the electrons move through this protein complex, energy is gradually released, and this energy is used to pump hydrogen ions, or protons, across the inner membrane, from the matrix into the intermembrane space. At this point, it might help to realize that moving electrons is electricity, so it should not be surprising that these electrons can provide the energy to drive these microscopic pumps made out of protein. They are basically natural electric pumps, created way before humans invented them, which is super cool. The electrons are then transported by this special red molecule toward their next destination. The red molecule also picks up the electrons donated by FADH2, that other electron carrier that we made during the Krebs cycle. All of these electrons are then passed on to another protein complex. At this next protein complex, the electron energy is used once again to transport hydrogen ions across the membrane into the intermembrane space. Notice that we are building up a high concentration of hydrogen ions in the intermembrane space. This will be very important soon. Now the electrons are transported to a final protein complex, where their energy is used once again to pump hydrogen ions into the intermembrane space, making the concentration even higher. Now the electrons need somewhere to go, a final destination to complete their journey. Fortunately, you breathe, and oxygen comes to the rescue. Oxygen accepts the electrons, as well as hydrogens from the matrix, making water. This is the reason why you breathe all day, every day. Your cells need oxygen to accept these electrons, or the electron transport chain will grind to a halt, like an LA freeway during rush hour. And if the electron transport chain stops, you won't be able to make enough ATP to keep you alive, and you die. So keep breathing that oxygen. Now you may have noticed that we haven't made ATP yet, so you might be wondering how that works, which brings up the coolest part of this entire process in my opinion. Remember that high concentration of hydrogen ions we've been building up? Now it's time to use it to make ATP. A protein called ATP synthase makes a channel that allows hydrogen ions to flow through from high concentration to low concentration. You may remember that molecules tend to move from areas of high concentration to low concentration naturally, a process known as diffusion. So, these hydrogen ions, in a manner of speaking, want to flow from the high concentration area of the intermembrane space to the low concentration area, the matrix. But they can only get through ATP synthase. This facilitated diffusion of hydrogen ions through ATP synthase causes ATP synthase to spin like a turbine and produce ATP. Such a cool mechanism. It's like a microscopic hydroelectric dam, generating ATP instead of electricity as hydrogen ions passively flow through. Since we made many electron carriers throughout respiration, the electron transport chain happens over and over again, making between 30 to 34 ATP molecules for every glucose that entered respiration.
So that was the chemiosmosis process. And as I said, we saw the same process in photosynthesis. So this is the main process that's going to manufacture the majority of the ATP in the cells. All right, so this type of ATP synthesis makes up to 34 ATP molecules from one glucose molecule. So this specific type of ATP synthesis, also known as oxidative phosphorylation, involves a proton or hydrogen gradient across a membrane. And there's also an electron transport chain for electron transfer. It takes place across the inner membrane of the mitochondria, and it also took place on the thylakoid membranes of the chloroplast. So essentially what happens is that these in the membrane, they represent electron carriers. And when they get, when the electron carriers, hydrogen ion carriers, when the carriers come there, electrons are passed along the electron transport chain, and energy is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. All right, so we pump hydrogen ions from the mitochondrial matrix into the intermembrane space, and there's a buildup of hydrogen ions in the intermembrane space. And then the hydrogen ions are allowed to pass through this special enzyme here, or this special protein here, which is the ATP synthase. So it is that they are allowed to go through ATP synthase, and as they pass through the ATP synthase, that is how the energy from the molecule is captured and is used to make ATP, right? So basically, it involves us pumping hydrogen ions in one area, and when the hydrogen ions build up, they allow them to pass through this special protein here, and that energy that they have as they're passing through is used to make ATP, right? So this same process, we saw it in photosynthesis across the thylakoid membrane. Now we're seeing it in the mitochondria, where we're pumping the ions from the matrix into the intermembrane space, and then we allow them to flow from the intermembrane space back into the matrix while passing through this enzyme here, the ATP synthase. You can see that the energy is used to combine ADP with phosphate to create the ATP molecules. All right, I don't think you need to copy the diagram. The diagram is really there to illustrate what's happening. I'm going to describe what the diagram is showing though. So this is the description I want to copy. We will meet this again when we go further in respiration. This is the last part of respiration, the electron transport chain. So when we come back to this section now, we can actually draw it that time. So we're basically building up a hydrogen ion gradient, and then we use that gradient, the energy from that gradient, to make ATP. And as we said, the majority of the ATP molecules that are made from every glucose molecule is done by this process. The substrate level only makes about 4 or 6 molecules. So remember, you don't have to copy this diagram. It's just there for illustration. So remember, we're not drawing the diagram, right? We're just writing the description. So I'm just going to finish up the notes now. All right, so describing the process now that we saw in the diagram. So after we build up, so the first part of it says that this specific type of ATP synthesis involves a proton hydrogen ion gradient across a membrane and an electron transport chain for electron transfer. It takes place across the inner membrane of the mitochondria and the thylakoid membrane of the chloroplast. All right. So first thing we should have said though, a piece of this is missing, right? So 2 points are missing. All right, so electron carriers such as FAD and NAD bring electrons to the inner mitochondrial membrane. As these high energy electrons move from protein to protein in the chain, their energy is used to pump hydrogen ions from the matrix of matrix into the intermembrane space. So there is a buildup of a high buildup of hydrogen ions in the intermembrane space. Let me read it. All right. Electron carriers such as FADH and NADH bring electrons to the inner mitochondrial membrane. As these high energy electrons move from protein to protein in the transport chain, their energy is used to pump hydrogen ions from the matrix into the intermembrane space. There is a buildup of hydrogen ions. There is then a buildup of hydrogen ions in the intermembrane space. Then, the notes would continue now to say that the protons will then move down their concentration gradient into the matrix. Because now there's a high concentration in the intermembrane space and they want to go back into the matrix. But for them to go back into the matrix, they have to pass through the ATP synthase complex. And the ATP synthase uses the energy to drive the phosphorylation of ATP. And the electrons that were traveling along the chain are going to be finally accepted by oxygen. And oxygen serves as the final acceptor of those electrons and for the protons, those forming water. Most of the ATP made during respiration is made by this process of chemiosmosis, which is about 32 to 34 ATP molecules per glucose. Okay, 2 more minutes, guys, and we're gonna be moving on to learning about the mitochondria, or the mitochondrion, which is the organ in which respiration is going to take place. And we are moving, as we know, we are on the topic of respiration right now. So we have to spend time learning about this organelle known as the mitochondrion, like what is its structure, its function, and how its structure is adapted to its function. So we're going to answer the structure of the mitochondria. All right, so I've tried to find simplest structure I could find for you, because this is something they expect to know how to draw. And I'm going to add some features to it. So this is the mitochondria. So we have the outer membrane, and then there is the inner membrane, which is known as the inner membrane. And then it's highly folded, and one fold is known as a crista, or two of them is known as are known as a cristae. Those are the folds, right? And then on the crista, you can find you can see the little enzymes, the ATP synthase enzymes that are on the inner membrane, on the crystal. Right, so I'm adding some detail to it. Yeah, you should. So I'm adding the other things that are not here, that are important. So in the matrix, which is inside, you see some black dots would represent the ribosomes. Other thing that you should add, so you should add some DNA, because the mitochondria has its own DNA. It actually has its own mitochondrial DNA, right? So I'm actually going to annotate this diagram, because most of the time they want an annotated diagram. So that means that I'm just going to give some functions beside the structures. All right, for example, the outer mitochondrial membrane is a selectively permeable phospholipid bilayer. Which encloses the mitochondrion and controls the entry and exit and molecules. So my attempt right now is to give you a description for each of the structures. So the mitochondria also has its own ribosomes, so it can actually manufacture its own proteins. The inner mitochondrial membrane now is highly folded. And contains the electron transport chain. And ATP synthase. I made it look circular, yes, because it actually looks like bacterial DNA, which is circular. Oh, the circle is fine. Um, Then we have the intermembrane space, which is the space between the inner and outer membrane. Area where we have a buildup of hydrogen ions for chemiosmosis. We have the matrix for the cristae, which are the poles. The poles is there to increase the surface area. So the matrix now has a gel-like substance. It's one of them. It's a mitochondrion when you have one. Mitochondria is plural. So technically, if we're looking at one, it's a mitochondrion. Oh, sure, that's what you mean. Outer mitochondrial membrane or inner mitochondrial membrane. We can say outer membrane, yes. The matrix has a gel-like substance, and it basically has the enzymes that are required for the link reaction and the Krebs cycle, which you will learn about. And the mitochondrial DNA codes for mitochondrial proteins. Yeah, so you have to know how to annotate a diagram of the mitochondria, which we just did. So the other thing to look at is how is this mitochondria adapted for its function? So I'm going to give you 5 more minutes, and then after that, we're going to look at adaptations of the mitochondria forward function, and then we see what pathways we can do based on what we covered today. So guys, are we ready for the adaptations now? Um, right, so as you go through the adaptations, you might see some similarity to what we did when we annotated the diagram. All right, so the mitochondria, the function of of this organelle is really to carry out oxidative phosphorylation. Right, so mitochondria are rod-shaped organelles, which are like 0.5 to 1 micrometer in diameter. And they are the site of aerobic respiration. So their function is to synthesize ATP. That's the overall function of the mitochondria. And then we're just going to see now how it is suitable for its function. All right, so the outer membrane is more permeable than the inner membrane. And this allows compartments to develop. For example, we have the intermembrane space and the matrix, which are different compartments within the mitochondria. Now the inner membrane now, the crystal, and the cristae, this causes a great increase in the surface area, which makes ATP production way more efficient. The inner membrane folds into the cristae, and that is what dramatically increases the surface area that's available for embedding the proteins of the electron transport chain and also the ATP synthase proteins. And this maximizes ATP production. So if the inner membrane wasn't so folded, then you would have less space to put in the proteins. So that's why it's so highly folded. So you get more space, put more proteins and more ATP synthase. All right, so another adaptation the intermembrane space All right, so the intermembrane space is narrow, and this helps to concentrate the photons that were pumped from the matrix. And this establishes an electrochemical gradient This establishes the necessary electrochemical gradient to drive ATP synthesis via ATP synthase. For the mitochondrial matrix now, um, matrix. All right, so the mitochondrial matrix is a viscous space that contains the enzymes that are necessary for the link reaction and the Krebs cycle. This compartmentalization keeps substrates and enzymes together for more efficient reactions. Next, we're going to talk about the DNA and the ribosomes that it contains. I said the mitochondria possess their own circular DNA and ribosomes, and this allows them to synthesize to make their own essential proteins, including enzymes for aerobic respiration independently. So they don't have to wait for the cell to make it. They can make their own proteins for themselves. So the last thing we have to talk about would be the fact that it has a double membrane system. right, so the double membrane system includes the outer membrane and the inner membrane. So the outer membrane so the outer membrane contains a protein which regulates the transport of substances such as pyruvate into the mitochondrion. And then the inner membrane is responsible for oxidative phosphorylation. And those would be the adaptations. All right, almost there. So the outer membrane regulates the entry of important substances into the membrane, into the mitochondria. But the inner membrane is where we have oxidative phosphorylation taking place. So the double membrane system allows us to form compartments essentially, where we can have specialized processes in different compartments. And it makes the entire organelle more efficient. All right, so I'm going to give you 3 minutes to finish copying that, and then we're going to see what past papers we are now able to do based on what we've learned today about ATP molecule and about mitochondrial structure. After 2 more minutes, guys. Right. So now what can we do in terms of past papers based on what we covered today? All right, so let's go to Canvas and let's see what you have to do. Guys, remember, this is a part of your class. It's optional, it's mandatory. All right, so if there's one thing you should know by now is that great work or high levels of performance comes from consistency. It's not about brilliance. It's about being consistent, right? So even if you have the talent for something, somebody who will develop the discipline will outperform the person who has the talent. So just get into the habit of doing things habitually each week, and you'll find that you achieve more without necessarily being the brightest. All right, so we are looking at respiration part 1, week 4. So there is a handout that talks about the structure of the mitochondria, structure and function. So you can read that handout for further reading. But here are the questions. So this 2019 question says that the mitochondria are important for making energy available within cells. Figure 2 is an incomplete diagram of the longitudinal section of the mitochondria, which only shows the outer membrane. Complete the diagram and include all the labels to illustrate the structure of the mitochondria. So basically, they only gave you the outer membrane, so they wanted to draw everything else for 5 marks. Explain how the structure of the mitochondria is important for its function. I think it's probably about, this was probably about 6 marks. How is the structure important for its function? So that part there, you're just gonna have to summarize the notes I just gave you, all right? How it's structured. Yeah, we can summarize the different parts, right? That's basically what it is. And then this is 2017, mitochondria play an important role in cellular respiration. In the box below, draw a labeled diagram to show the detailed structure of the mitochondria. 3 marks for that. So notice that these diagrams, they did not ask for annotations. So I give you the annotated one, but that is not what you're asking for here. You just want the diagram and for it to be labeled. All right. On your diagram, use labeled arrows to show 4 key substances of cellular respiration that must come out of the mitochondria. All right. Things that leave the mitochondria would be ATP, because that's what it's making. Water, because whenever the oxygen accepts the electrons and it combines with hydrogen, you get water. Also, NAD+, FAD+. So the NADH and FADH were the ones that brought hydrogen and electrons to the mitochondria. Once they deliver their electrons, then it goes to being FAD+ or NAD+, and those need to leave the mitochondria to go back to picking up more electrons. So that will give you like 4. Now, so they're just saying show 4 substances that are leaving out of it. So just draw, after you draw it, you'd have drawn an arrow pointing out and then list 4 substances. Yes. This one now from 2014 says this is a structure of a part of the mitochondria seen through the longitudinal section. So they labeled A, B, C, D. All right. So D actually is a DNA. So this one they do draw it coiled up. So what does structure C look like? So these would have to be the ATP synthase, right? Along this cristae. B is referring to the cristae or crista because it's just one. And A is pointing to the matrix. So they're like identify these structures and use an arrow labeled X to indicate where the Krebs cycle occurs. So the Krebs cycle occurs in the matrix. All right. So these are the questions based on just mitochondrial structure. All right. So this is for classwork, guys. So this is something that you can more than complete and get submitted on time. All right. So in our next class, we're going to go deeper now into the process of respiration because we haven't started that yet. We're just doing the background. Today we spoke about ATP the immediate energy currency and structure the mitochondria. So next week, now we're going to break down the stages of respiration, which would be glycolysis, Krebs cycle, and electron transport chain. So we're going to do stage by stage for the process of respiration. All right. And that's where we stop today, guys. Enjoy your day, and please make sure the classwork is submitted on time. Okay? All right, that's it. Bye, guys. Thank you.